name's Mia. So I'm a postdoc in May Zen's lab at the University of Toronto, and I'll talk a bit about our journey into volume EM for connectomics. So back in the 70s and 80s, I think most people know the story. John White and colleagues spent 15 years mapping the mind of a worm in a really backbreaking, manual, labor-intensive process. And it's formed the foundation of C. elegans neuroscience. But everything's much easier now. So Mia just gave this great intro to high-pressure freezing. So we can get a really nice sample that's fixed in a way that is as close to the native ultrastructure as possible. So things are much clearer when we try and look at them. And microscopes have gone through huge advances. So nowadays we have digital cameras. Rather than having to develop films, we have these new generation scanning electron microscopes, which are really nice. They have these backscatter detectors that get you lots of signal really fast. And we have automated imaging approaches, so we don't have to physically be there in the microscope room while we do it. And obviously, modern computing has come on leaps and bounds, and this really helps all the way from taking our images to stitching them into a 3D volume and going through and annotating your micrographs. So when we started out, we started using serial section TEM. So basically, we cut our ribbons just like Mia showed, and we collect them on these slot grids. These slot grids have a piece of plastic film going across it, and they support the sections. And you can see a little worm in each of these sections here. And you fire electrons down, they get deflected by the worm, and you have your images. And this worked really great for us. It's the same way that Nicole Thompson did it back in the 70s and 80s. And it's a great option because if you have an EM lab in your university or your institute, then they very likely have everything that you need to do this. You don't need to buy anything special. If you talk to your EM core about doing some C. elegans EM, there are a couple of pointers that you might want to give them to make life easier for you in the long run. One Mia just alluded to was the orientation of the worm is super important. You want to be able to cut it either transversely or longitudinally along the ventral dorsal axis so that the worm isn't going in and out of the section. And it just helps to be able to ID things and see where you are. And you want to use slot grids rather than mesh grids. So this is a slot grid with the sections supported in the hull. Mesh grids support the sections by having these copper bars that go in a mesh along the grid. And you will find that, I guess it's a part of life, you'll always end up with your worms over the slot bars, over the grid bars. And in C. elegans, it's important to try to avoid that because everything's so small. So if you miss a few sections, it can actually have a big impact on your data, especially if you're trying to look at the nervous system. So another way to go from a plastic block to your images is using scanning electron microscopy, where you bounce electrons off the top of the sample. And I mentioned there are these really nice new generation scanning electron microscopes with these really nice detectors. This is what we've moved to in our lab now. So we use Irina's variant of array tomography SEM. So we have this 4x3 centimeter silicon wafer. And I cut a whole worm onto this wafer. And we can go put it in our microscope, go to the ribbons and sections, and we basically just draw boxes around our region of interest, spend a couple of hours doing that, then click play and go home, and the microscope does all of the work for you. So when this was developed, this was originally developed for multiplex labeling or correlative light and electron microscopy. So this was developed by Christina Mashiva and Stephen Smith. And the idea was that you take your tissue, you cut your sections, put them on a glass slide, and then you can stain for your antigens of interest, then strip it, then stain it, and so forth. And then after that, you can stain it with heavy metals and look at it in the electron microscope. And this has been used to very nice effect by Irina here in this Burra et al. Paper, where she did 2 rounds of staining using 4 antibodies and DAPI. So you can get this really nice molecular information on top of your ultrastructure. You can also combine it with super resolution microscopy, like Sebastian did here when he was looking at gap junction proteins in the nervous system. But we don't really do the molecular information yet because it requires some very specialized free substitution. And so when we fix our samples, we fix them specifically for the ultrastructure so that the ultrastructure is as clear as possible. And using this technique, we get these really nice volumes of worms that we can use for our kinectomes. And lastly, another way to get your block into images is to section it using an atom, which is an automated tape collecting ultramicrotome. And so you section it like normal into this diamond knife. The sections float on the water, and they're collected by this roll of tape that just dips down into the water and gets rolled up. And then once this is finished, you just cut your tape into strips, you stick it to a silicon wafer, and you can image it in the scanning electron microscope. And we've had great success with all of these approaches. So one more consideration when you're starting your experiments is to think about what kind of thickness you need in your sections. So when we started trying to use 70 nanometer sections to map connectivity in the nervous system. And this ended up being quite tough because we have 30 nanometer vesicles, so in one section you can have 2 vesicles on top of each other, and it's kind of hard to map the connectivity like that. But if we move to 50 nanometers, everything's just so much clearer. You can see these beautiful synapses, these vesicles, etc. And then if you have a super stable ultramicrotome and you can go to 30, it's even better. But just because it's more stable, we tend to stick with 50 nanometers. And Mia already mentioned TrackEM, which is what we use to stitch our images into a volume. And it's just a Fiji plugin. It's super accessible. This is not the nervous system. This is spermatheca. It's 300 sections. Acquired it last week, and just using the default parameters of TrackEM with about 10 minutes of active clicking and a few hours of waiting, we've already got it into quite a nice volume. So it's probably got a few more hours to kind of fix some of this jitter, and then it'll be ready to annotate. So once you've got your EM volumes, you need to go through and identify the cells. Luckily we have this great map from John White and colleagues. One feature that we can use to identify our cells is the position of the nuclei. And this tends to be relatively stereotypic, especially, so you've got the position within the worm, and also the position relative to the rest of the cell bodies. But there is some variability there as well, and especially in places like the retrovesicular ganglion. So in addition to that, we can look at the neurite trajectory, whether a neurite goes out anteriorly or posteriorly, whether it goes through a commissure. For example, this commissure always contains the same 4 neurons. But there is also occasional variability, and especially in mutants as well, which can do some weird things. And then the mind of a worm has these really nice neuron maps. So we have a cell body, and it projects around the nerve ring. This is AIB, then it moves into a new neighborhood, and then goes around, and there's a nice description of it as well. And Worm Atlas has this really nice clickable interface where you can go through the different neurons. So when I'm IDing neurons in a volume, I basically have this open on one screen and the volume open on the other and go through. The cells tend to, the neurites tend to take up very stereotypic placements within the bundle. So for example, the motor neurons in the nerve cord tend to stick around here, with DD GABAergic neurons coming here and sending little spines down, BDs here, and then you have the excitatory motor neurons a bit further down that send neuromuscular junctions that point up towards the muscle arms. And then you have the proprioceptive processes of the rest of the motor neurons that tend to sit down here. And then you have super stereotypic neurite morphology. So for example, RMDR is making this characteristic sheet-like process here in the ventral side of the nerve ring. And there are many of these really characteristic features around the nerve ring that you can use to help ID your cells. Then there's the texture of the cells and the intracellular features. For example, body wall muscles always sit in this area of the nerve ring and they're full of ribosomes. And then there's connectivity. So there are very stereotypic connectivity motifs in the nerve ring or the nerve cord. One of the most is most neurons tend to make a gap junction either on the dorsal or the ventral midline. So if you think you've found ROBL, you trace it round and it makes a gap junction to ROBR, you think you can feel pretty safe that you've done it right. So we have all of this information that we can combine to ID the cells in the nervous system, and provided you've got a big enough volume to fit these features in, then you can identify everything with 100% confidence. And you do occasionally get tricky cases. In that case, you just stop, move on to another neuron, and you kind of help to figure it out by exclusion of other neurons. So we do see some variability in the nervous system as well. So for example, earlier this year I was a bit confused because there was a worm with an extra neuron. So it seems like there are two RICRs. The cell body was in the same position. They sent these processes going in the same area and making the same connections. But it turned out that RICR has a sister cell that's supposed to undergo apoptosis. And it looks like in this case it just simply didn't. And it took on RICR fate. So we've seen this a couple of times in our EM volumes. So one thing to pay attention to when you're beginning your sample prep is to stain your tissue so that it highlights whatever you want to see. So for example, we're looking at chemical synapses and gap junctions. And we have this staining condition which is quite faint around the cell membranes here, but you have very dark chemical synapses that are very easy to see, and the gap junctions are selectively stained as well. So this means that with this staining we can go through our volume and get quite a high confidence readout of connectivity. But this staining condition isn't so good for someone who's interested in mitochondria, for example, because you can't really see the mitochondrial cristae very well. So in that case, you just use a different protocol that was optimized to highlight those kind of features. So we have quite a few data sets so far, many of which are already on BOSDB. There's also a Christiankis data set on BOSDB, and I hear Worm Atlas are also going to be getting their data sets on there as well. So it's a nice place to go and take a look around, and we're hoping to get some more data on there soon. So in terms of the timeline for doing this, so high pressure freezing for substitution takes a couple of hours. To do the high pressure freezing itself, then it can be from a couple of hours to a couple of weeks to do the freeze sub. Embedding, as Mia alluded to, can be quite frustrating, but it can take a few hours to a week. Sectioning, we can do in a day. Imaging, adult nerve ring using this serial section array tomography takes about 1 to 2 weeks. You can stitch it into a nice volume in a few days, and it takes about a month to do a round of synapse annotation, and a few months to color everything in and get a 3D volume. So we have these bottlenecks currently, which are the 3D segmentation, where we literally have a pad that we color things in, and this is being sped up using machine learning. And Alex Bay, who helped with the Mind of a Dower, which we've heard about at this conference, also has a really nice way to use machine learning to accelerate chemical synapse annotation. So in summary, I think I'm running out of time, but there are all these approaches that you can use to get your volumes, and thanks to everyone who was involved in developing the pipeline, and thanks to our funders. Thank you.